Appalachia this month, and I'm going to introduce Dr. Rebecca DuBois. She's going to talk about bioterrorism and bioterror agents. I think this talk is particularly relevant since there's all those things that have been happening at the CDC. Like they just found vials of smallpox that they didn't know about in cold storage, and a variety of other things that have been happening. But I want to introduce Rebecca. She's an assistant professor in biomolecular engineering at UC Santa Cruz, and she's a structural biologist studying proteins from human viruses. She uses her discoveries to design new vaccines, develop antiviral drugs, and engineer nanoscale drug delivery. Before joining UCSC, she got her PhD at UC San Diego in biochemistry, and she also did a postdoc at the Pasteur Institute in Paris, and also another postdoc at the St. Jude's Children's Hospital in Memphis, Tennessee. She has also received a very prestigious award, the K-22 Research Scholar Development Award. It's for professors early in their career, and it's a very difficult award to get. So I'll leave it up to Rebecca now. All right. Can everybody hear me?
Yes.
Yes. All right. First, I want to say thank you very much to the Women in Science and Engineering. They are a fabulous support group for all students at UC Santa Cruz, and they put on awesome events like this. It is It's a rare treat. I was saying to my husband that I get to drink beer and give a talk to beer drinkers. We don't normally get this at science conferences. And I also want to mention to any terrorists out there, I'm not giving up any trade secrets. So if you're looking for some clues for the next bioterror weapon, not getting it here. But I will tell you about bioterrorism and the history of it, and what are the perceived current threats that could be used in cases of war or in our country and over the world. And then at the second part of my talk, I'll be talking about the ethical considerations that researchers studying potential bioterror agents, the ethical considerations they have to actually do that research and publish that research. So first of all, what is bioterrorism? It is an attack, a deliberate release of viruses, bacteria, or other germs. I'm going to sort of call them bioterror agents. They could be just biological toxins, not an actual infectious particle, but a toxin that could cause illness and death. We usually just think of people, but they could also be used against animals, livestock and poultry, or in plants, or in our water supply and food supply. So bioterror agents could be dangerous in a number of situations. So I'm going to give you a little history. It is not a new thing, the use of bioterror agents, especially in terms of war. So back in 600s BC, the Assyrians poisoned wells of their enemies with rye ergot fungus. Sorry, ergot. Seen right here. And when their enemies ate this, they got convulsions and gangrene. So they weakened their enemy by poisoning their food supply. In the 1300s, the Tartar army put plague-infested corpses over the walls of a city, the Kaffa city they were trying to dominate, and it caused an epidemic. The city had to surrender, and it's thought that that was the start of the Black Death in Europe. And so there is a historical precedence for the use of bioterror agents. Sorry. All right, and in the 1700s, during the French and Indian War, the British gave blankets to Native Americans, and these blankets were used by smallpox victims. And smallpox is spread through contact with the skin of other smallpox victims. So the Native American population was really decimated by smallpox. More recently, in World War II, Japanese launched many experimental attacks of anthrax, shown here as anthrax spores, and plague, which is spread by, or plague-infected fleas, and cholera. They dropped bombs or sprayed gases of these agents and killed thousands and thousands of people in China. Around that same time, USA launched a bioterror weapon development program. So our own country was developing all sorts of really terrible bioterror weapons, especially anthrax bombs, shown here. And however, I think we realized that this was going down a very dangerous path, and we could be not only killing our enemies, but the whole entire world by developing these kinds of things. So President Nixon signed an order to stop that development. And they got a lot of other countries in the world to sign that as well. And for those of you that remember, in 2001, several letters that were laced with anthrax spores were mailed to several people, U.S. senators and news agencies, and ended up killing a lot of people and infecting people. And that case, in my mind, is still not completely solved. The person that was sort of the main suspect committed suicide, but from what I've read, I'm not even totally convinced that was the right person. They were just badgered so much they committed suicide. So I think it's still open for question. It's really interesting, all the evidence for this and what was the motivation for this. But because of this act of bioterrorism with the anthrax letters, President Bush signed a Bioterrorism Act, and that really was established to prepare ourselves for a potential bioterror attack, to establish a preparedness plan, to enhance controls on dangerous biological agents and toxins, ways to protect our food and drug supply, our water supply, and other things. And at the time, I was at UC San Diego in graduate school, and we studied a bunch of different bacterial pathogens in the lab. And we basically had people from the CDC come in and raid our freezers and take out anything that we weren't allowed to have anymore that they now made considered dangerous biological agents that they wanted under complete control. So there was really a lockdown on these biological agents. So how do we assess what are the biggest bioterror agents out there? If you can imagine, there's tons of things that cause diseases. You could potentially engineer them to be worse in some way, and so a group of a number of different agencies, the Center for Disease Control, NIH, and Department of Homeland Security came together and came up with some guidelines, what defines a bioterror agent and its potential. And number one, they asked how easily can a bioterror can an agent be spread? And one way to think of this is dissemination. Can it be distributed to many people via air or water supply or food or direct contact? Or is it highly transmissible, meaning once it infects a couple people, it spreads really rapidly from person to person? So two different ways that an agent can be spread. And next, what is the severity of illness that this agent causes? How pathogenic is it and how virulent is it? And those are two different things. One is pathogenic, the ability to cause a disease. And as scientists, we define that as the number of particles required to cause an infection. Some things are so infectious, one single particle, one virus or one bacteria is enough to cause complete disease in a human. But that doesn't necessarily mean that they're going to die. So another way to look at the severity of a pathogen is virulence. And so how many particles are required to cause death and mortality. So if you look at all these, the ability to spread and the severity of illness, the CDC and NIH have come up with a list of bioterror agents categorized as A, B, and C. And category A agents are what are perceived as the biggest threat. that they can spread very easily. You can get your hands on them easily, and they could cause major harm, illness, and death. And so they could also cause public panic, social disruptions, and they require special action and preparedness. And so here is the list of 6 different Category A agents. The first one we know about, anthrax. It's found in nature. It's very easy to disseminate. Even back in the 40s when we were developing bioterror agents, we were able to develop anthrax bombs, no problem. One that goes off in New York City. If you couldn't get antibiotics to all those people that in New York City within the first 24 hours, you would see major death and destruction. So we not only need a plan to be prepared for that, but a plan to be prepared to, I mean, for anthrax, if you get infected, you just need antibiotics right away, but we need a plan to get those antibiotics to so many people right away and to stockpile those antibiotics. The next one is botulism. It is found in contaminated food, and it is on the Category A list because 1 microgram, that is 1/1000th of a milligram, which is about the weight of a paperclip, 1 microgram could kill a human. So just an invisible drop, when ingested or put on the skin, could kill a human. So the botulism toxin is very potent. The plague is also on the list. It has a very low infectious dose number, so not too many bacteria are required to cause infection. It's spread through fleas, and it's highly fatal if you get infected with it. Smallpox, which is eradicated from the world. We have an excellent vaccine, but all the vaccination programs stopped back in the 1970s because we eradicated it. As you probably heard in the news, some vials of it were found recently. I think, was it at CDC? At the CDC. So where else could that happen? You know, where else could these vials be found from researchers from a while ago that were stored away in some freezer and forgotten about? So I think it could be a very real problem. Anyone born after 1970 has not been vaccinated, so that's a huge percentage of our population that's not protected against a potential smallpox bioterror agent. And smallpox is one of those cases where only one virus, one single virion is enough to cause an infection. Tularemia, also a really bad infectious disease, has a low infectious dose and highly fatal. And then hemorrhagic fevers. We've heard a lot about Ebola in the news lately, certain cases of Ebola, and concern that it could come to the United States. And it is highly infectious as well. Between 1 and 10 virions only are required to cause an infection, and highly fatal, up to 90% fatality. So these are the worst of the worst in terms of bioterror agents that could cause real devastation. And so I'll do a case study on botulism later on in the talk. The second category in bioterror agents are high priority still because they're moderately easy to spread. They still cause illness, maybe not high death rates, but still can cause death and disease. And they require enhanced disease monitoring. So we're still looking in our animal and supplies and food supplies for these agents. And they consist of food safety threats like salmonella and E. coli, water safety threats like Vibrio cholera, a number of agents that can infect livestock. It would be really devastating if all our livestock were wiped out. Similarly, if all our poultry were wiped out with a poultry bioterror agent. So these are also highly watched by our government. And third category agent are ones that are perceived as potentially bad, but right now we have them under control. But because of their ease of production, their ease of dissemination, they can be spread from human to human or easily disseminated. They have potential and so they They are perceived threats. There's no direct threat right now, but they are perceived threats that they could be engineered for mass dissemination. A number of tick-borne viruses, yellow fever spread by mosquitoes, I believe. I'll be talking about influenza virus, rabies. You see this dog has rabies here. Tuberculosis, prions, and down here it says SARS and MERS. You guys have probably heard about those in the news. Those are perceived, major perceived threats for bioterror agents, but right now not necessarily seen as a major bioterror agent. Okay, so now you've learned all about the really bad bugs out there. And the next part of my talk, I want to ask the question to you guys, and I want you to think about this. Should scientists and researchers study these bioterror agents, or should we leave them alone? And if we study them, should we be publishing our results on that research? And I'm going to be talking about 2 case studies. The first question, should scientists carry out research? The research could advance the knowledge and development of cures for these agents, but the perceived risk, especially in the public, and especially with the media lately and things like the smallpox vials, there is risk of escape of that bioterror agent from the laboratory itself. And then the second question, should scientists publish that research? Scientists see publication as a fundamental thing to scientific advancement. We need to share that information. And it's a form of freedom of speech, too. However, if we publish that, that information could get into the hands of the wrong people, and that knowledge could be misused for development of a bioterror agent. So there's this balance, this risk of public health and benefit versus public security and safety. And this is essentially the definition of what's defined as dual-use research. That is scientific research that can be used for both good and harmful purposes. So scientists aren't bad people trying to destroy the world. We're legitimately trying to understand these pathogens and find cures for them, understand them. And however, that information could be misused and cause harm. A classic example of dual-use research is nuclear research. If you think about it, scientists were just studying nuclear fission as a way to produce large amounts of energy. And we've developed nuclear reactors. We have 65 nuclear reactors that generate almost 20% of our country's electricity. However, nuclear fission was also a way to make an atomic bomb. And in 1945, 2 atomic bombs killed 200,000 people instantly. So this is a classic example of dual-use research that was designed for the benefit of humanity, but used for ill purposes. So I want to tell you about, I think it's important to let you guys know that researchers can't just study these bioterror agents anytime and anyhow that they want. This research is highly scrutinized and regulated by our government. Led by the National Science Advisory Board for Biosecurity, provides guidance to the government and several funding agencies for researchers on dual-use research. And so this is the typical approval process to carry out a dual-use research thing. And so first it starts with the science. Every grant application is peer-reviewed. So your scientific peers have to agree that this is important research that should be done. There's the Office of Biotechnology Activities, the Biosafety Committee, Animal Care and Use Facility, if you're going to use animals in your experiments. And then, really importantly, you have to have facilities to do this work. These agents are usually studied in biosafety level 3 or 4, which requires the full hazmat uniform, fully contained, airtight facilities where you don't even breathe the air of that facility, and the agents you're working in are in its own separate container where you're using the spacesuit hands to kind of manipulate your samples. So these are no run-of-the-mill research facilities. These are highly controlled. We don't have a BSL 3 or 4 facility at UC Santa Cruz. But in addition to these facilities, there are regular site visits, multiple inspections, and the people that get to use these facilities go through rigorous background checks. I've known colleagues that have waited over a year to just get approval to do the research that was already funded and approved, but they themselves had not gone through the screening process yet and needed to be fully documented with background checks. And finally, there needs to be approvals to transfer the material, the biomaterials, from one place to another. Okay, so first case study I want to talk about is a Class A bioterror agent, botulism toxin. Botulism can be, you can get infected with botulism by eating contaminated food. As you can see here, no matter how hungry you are, do not eat soup from a can that looks like this. It likely has botulism.
You're scaring the grad students.
Don't, even if you're starving grad students. So botulism is caused by a spore-forming bacteria, Clostridium botulinum. There are not too many cases a year in the United States, about 150. 65% are infant botulism that's just obtained from contaminated soil or contaminated honey, which is why you don't give infants honey. Apparently they just don't have the digestive system mature enough to handle that, but adults do. There's also foodborne botulism, improperly canned food, and wound botulism, usually from injectable drug use. The symptoms are blurred vision, drooping eyes, and slurred speech, but this progresses to paralysis, usually in the respiratory muscles, and you just don't have the muscles to breathe. Again, it's the most potent toxic known. A microgram could kill a human, inhaled or injected. There's a number of different types. We have anti-sera to types A through G. And the toxin works, I'm not going to go into the details too much, but they block the signal called acetylcholine, the signal that the nerves use to tell the muscle to contract. So if they're blocking that signal, your nerves might be trying to work, but they're not connecting to tell the muscle to work. So the only treatment are breathing machines and antitoxins, which don't reverse it, but they'll block its progression, so if you catch it early enough. But it's not all that bad, because botulism toxin has given us Botox. As you can imagine, if you can't have muscle contraction, you won't have wrinkles. And so extremely, extremely small amounts of botulism toxin are injected into your wrinkle lines, paralyzing those muscles and preventing that wrinkle to form. But it's not just for cosmetic use. Botox is also used to treat neck spasms, excessive sweating, which I don't fully understand the mechanism for, overactive bladder, crossed eyes, and migraines. So I just want to tell one little case study here about botulism. So recently, this was just last year, there was a single case of infant botulism, and the doctors found that none of the existing antitoxins were working, either alone or in combination. And they realized it was a completely new strain, type H toxin, and it was the first new type of botulism toxin identified in over 40 years. So it was a real surprise to find this. So my question to you guys is, should the genetic sequences of this new strain be published? All in favor? All the scientists in the crowd. All not in favor.
I'm not sure.
If you know the sequence, the ability, the information to make it in the lab is already out there. Right now we can already make those botulism toxins, but we have the antitoxins for those. So should this be published? So the tests that confirmed that it was a new strain of botulism toxin were published. However, the genetic sequence was not disclosed. And this is one of the very few examples of censorship in scientific research and information. However, the authors do say that once an antitoxin is produced, they will disclose that. So that was self-censorship by the authors, the researchers themselves. And I think it was the right thing to do to protect, that information could be really devastating otherwise. And then I want to tell you about one more case study. The Class C bioterror agent influenza virus. I studied influenza virus for a number of years when I worked at St. Jude Children's Research Hospital. First, I'll just tell you that influenza virus, we've all probably had the flu. It is not fun. Causes fever, muscle aches, vomiting, diarrhea. It's actually transmitted, despite it feeling like a stomach infection, it's transmitted through the respiratory, through the air. Infection is in the respiratory tract. And in fact, humans are regularly only infected with 3 different types, H1, H2, and H3. But there are 17 different types of influenza virus, and they're found in our wildlife, especially in wild birds, wild migratory birds. And so if you've heard of H5N1, that's the bird flu, that is an epidemic right now in our migratory birds. And when they don't get the disease though, but when they interact with poultry, the poultry are susceptible and die. Rarely there is infection from, say, poultry to humans, but usually what happens is that swine are a mixing vessel. And so if you remember the H1N1, the pandemic swine flu, that was a mixture of a human and bird flu that happened in pigs and resulted in a pandemic. So what happens in a pandemic, it can happen one of 2 ways. There's something called antigenic drift. And so the virus just slowly mutates over time. And so it looks a little bit more different sort of every year. This is why we have to get the flu vaccine every year, because we're vaccinated against this strain, but it starts to look different over time. Then there's something called antigenic shift, where 2 different viruses mix together and make a brand new one. And so we are not able to recognize that brand new one. And so both antigenic drift and shift have resulted in pandemics in our history. The worst one was the Spanish flu in 1918 that infected most of the world and killed between 20 and 50 million people. Most of them were the healthy aged people. It wasn't the older, the younger. That occurred by antigenic drift of a bird flu that slowly drifted and was able to infect humans and then go human to human. There was the Asian flu and Hong Kong flu. And more recently, as I mentioned, the swine flu. And right now we have H5N1 and H7N9, which are seen as pandemic potential viruses, meaning they're highly fatal, but luckily there's no sustained human-to-human transmission. But the question is, okay, occasionally these bird flus do jump into humans, and they're really deadly. How close are we to those H5N1 influenza viruses going from human to human? What does it take? And so you can see right here, in Asia, there's a lot of interaction between people and poultry in their markets. And so what is the chance that this is going to start spreading from human to human? So to answer this question requires a dual-use research project. What does it take to mutate a bird flu virus and make it go from mammal to mammal? How many mutations does it take? The pros to doing this study are that we'll know what mutations to look for during surveillance. We have highly sophisticated surveillance all over the world looking at sequences of bird flu, especially in Egypt and Vietnam, where bird flu cases are really happening. And this will tell us how close we are to the antigenic drift pandemic. And we could develop vaccines based upon this exact strain. But the cons are that bad people could take this information and make a pandemic instantly. The technology is already out there to make any flu strain that you want. So should this research be done? If so, should it be published? And what I'll tell you is that it was done. I'm not going to go through the details, but what scientists did is they put pressure, sort of lab-induced evolutionary pressure, to see which mutated viruses could transmit from a mammal to another mammal. Ferrets are the animal model for influenza virus. And they were able to isolate viruses that go from mammal to mammal. Influenza, H5N1 bird flu viruses, they were able to mutate. And what they found is that only 4 mutations are able to change a deadly bird flu virus to go from a bird to a mammal to a mammal. And in fact, if you look at the surveillance data, 2 of those mutations have already been found in circulating bird flu viruses in Egypt. So that means we're only 2 mutations left to a bird flu pandemic. And as you know, influenza virus is just one of those viruses that easily mutates. So the advancements and conclusions from this research are that we can now know what mutations to look for in our surveillance and monitoring. We can quickly cull poultry that have been infected with any kind of viruses that look like this and try to develop vaccines against these strains. But once the word got out that these studies were done, there was a media frenzy. People worried that scientists were just engineering bioterror agents for fun and that terrorists could use this information. This was before it was published. But the researchers sent their results to the National Science Advisory Board for Biosecurity, and when they looked over all the information, they decided that the authors should not publish this research, even though it was government-funded research. But they concluded that they should publish the outcomes, saying that they found mutations. It was this number, 4 mutations to make it transmissible. But they should not include the methodology and other details that could replicate the experiments. And this, of course, caused outlash in the scientific world about censorship and censoring life-saving science. So there's this tug and pull about the cost-benefit of this type of research. The international community really weighed in on this, and ultimately, after several months of deliberation, the recommendation was to publish this research in full, but to impose a moratorium, meaning a halt on any more of this kind of transmission research. Do you have a question?
Yeah. So if you can't openly publish your results, is another lab able to contact you and ask for your results if they're also approved by the NSABB?
I'm not sure what kind of censorship was imposed at that point, if they could share it with well-known labs that already study those kind of agents, if it was just sort of secret. In fact, the results were already presented orally at a scientific conference, so it was already kind of out there, and so that was part of the deliberation too. Like, it's already out there, you're really, impeding a lot of things by not publishing it. So that was sort of a major argument to just go ahead and publish it. And so there are the 2 decisions to finally publish it about 1 year ago. And so I just want to summarize by saying that I believe bioterrorism is a real threat, and it's important to weigh the pros and cons before carrying out research or publishing this data. And I just want to give one little mention to my lab. I'm just finishing up my first year at UC Santa Cruz, so I don't have a lot of my own research to tell you about. But my lab studies the molecular mechanisms of human viruses, and so we're trying to understand viruses at the molecular level to use that information to develop vaccines and new antiviral drugs. But we don't have to work with any of the viral agents. We engineer just parts, little proteins of the virus alone. So here's my wonderful lab, and I want to say thank you guys for listening. I'll take questions. Yep.
Were the 4 mutations just like point mutations, single amino acid changes, or?
Yep, they were single nucleotide, single amino acid changes.
In the same protein? I guess I can look it up, but.
Yes, mostly in one protein, one mutation in another.
To what extent are research lab institutions around the world actually aligned on that particular way of sort of like constraining research? Like, are there certain labs that are like, that's great, but I'm going to do research anyway?
Yeah, the question was how aligned are researchers around the world in terms of doing this research? And it's highly debated. Actually, one of my former advisors, my advisor from France, wrote an article, coming out against it, saying it's just too dangerous, we don't know the implications of it, and we shouldn't be doing this level of research. So it's highly debated still. I would say most researchers tend towards the do it and get that information out there. It's advancing science as a whole, but you still do need some major sophisticated equipment to produce or grow a lot of these bioterror agents. But it's still debated even in the scientific community. Yeah.
I feel that the whole technique with choosing to only publish the details after we've sort of identified some sort of antitoxin or some sort of combative agent towards that, is that a good sort of method? Maybe they could try to do those things side by side prior to publishing so that they can get the information out there, but it's not as, I guess, volatile or harmful to the greater public, or is that totally unreasonable and way more difficult?
Yeah, the idea to do that research, but at the same time push forward the production of a cure, a vaccine, or an antitoxin, and that's certainly a major focus for most researchers, is finding that antitoxin or that cure. What was not really publicized in terms of that influenza research was that they were using strains of bird flu that were susceptible to Tamiflu, our best flu medicine drug. So although it's still dangerous research, we have a drug to stop it. Our antitoxin, we've got a drug to stop it. So they were doing it in the safest way possible. There are plenty of bird flu strains that aren't that way, so they chose it in the best. They selected their experiment in the safest way possible. Yes?
Let me make two comments and then get your response. Bioterrorism is two words, bio and terrorism. And let me address terrorism first. I don't think you're going to stop terrorism by stopping a particular weapon. For instance, an airline is a dual-use invention, okay? Mm-hmm. And that has caused more problems in the United States than atomic bombs have in the United States. Mm-hmm. So, if you want to stop terrorism, you need to stop the root cause of terrorism, not a particular technique. Now, let me go to bio. Because if something can be discovered, it will be discovered. And if you put handcuffs on yourself, somebody else will discover it.
Yes.
In the Ming Dynasty, China led the world in science. But 400 years ago, it stopped dead. And the result of that was terrible insults and suffering to Chinese people. And I worry that if you stop research, because America is afraid of terrorists, you'll end up with a third-class, third-world country.
Yes. Yes. Did everybody get that? I'm going to summarize by saying he notes that bioterrorism is two parts, bio and terrorism. And first he addressed terrorism and said that terrorism is going to happen no matter what. You need to stop the terrorists. And a lot of things could be used as a terror agent. One example is an airplane. That is a dual-use product that could be used for good and harm. And the other address was bio, and the same thing that biology and science is going to push forward. If you censor it, someone else is going to discover it anyways. So he's against the idea of censorship as well. Any other comments or concerns? Yes?
This is just a comment. I was working in Tanzania in '85 for the Danish government, helping farmers with vertebrate pest control. And they sent me up to a bubonic plague outbreak in northern Tanzania to try and do something about it. All we could do was simply give them a lot of rat bait and take our tetracycline tablets that night. I had some beautiful slides of a lot of buboes.
Oh man. Wow.
Most people don't realize here that we have bubonic plague in California above 5,000 feet in the mountains, Sierra
Really?
Nevada.
Really?
That's right.
It's in a lot of the rodents.
Yeah, it's in the squirrels and ground squirrels
Ground
and chipmunks and all that. Yeah.
Wow.
I'm a wildlife biologist.
Wow. Ground squirrels above 5,000 feet, even
Yeah.
here in California, are rampant with
Yeah.
bubonic plague.
Nobody knows. It's above that, right around that area.
Don't
Higher
let
up,
them
not
live in your
down.
house.
Yes.
Yes.
I was just going to say that people who lived through World War II here became very aware of botulism, something that they hadn't been too aware of before. A lot of people had victory gardens and did their own canning. And green beans particularly were deadly. Because I'm not sure just why green beans were more susceptible, but there were a number of deaths from botulism because people did not properly can their green beans, and this became a big thing. We all learned about botulism.
If you're canning your own fruits and vegetables, do it correctly. Especially green beans apparently are the worst in terms of getting botulism in your can.
I think that's when pressure cookers became really
Yes.
So I was at a Midway's atoll in the northwestern Hawaiian Islands several years ago when we had a botulism outbreak in some of the birds. And I was just wondering, I mean, you talked about how like flu can move between birds and humans. Are there different strains of botulism that can move between? When they originally weren't sure what it was, while these birds were dying, they had to wear the hazmat suits.
Huh.
Really not fun in Hawaii.
That was in Hawaii.
In the northwestern Hawaiian Islands.
In northwestern Hawaiian Islands, there was botulism outbreak
they
in
figured out what it was, and
birds.
so they didn't have to wear the hazmat suits. But
Well, botulism is sort of a soil, found in soil, and that's how it can get into your foods and things. So I'm guessing they got it from the soil for some reason.
yeah, it was interesting to watch them clean it up.
Mm.
You had to keep the birds from going in the infected ponds and stuff.
Oh, how do you do that? Some big nets?
Well, first we were chasing them around to keep them away from these. And these are endangered birds.
Okay, well, you're wearing a hazmat
No,
suit.
by then they've given up on hazmat things. But
Okay.
we're chasing them away from the infected ponds, like at night, running around. And then eventually they figured out that they could just put big lights up. Unfortunately, we had big lights because there was runway and stuff. And that would keep the birds away at night at least. And then they could clean it up through the night. It was an experience.
I didn't quite get all that, could you?
So apparently there was a botulism sort of outbreak in wild birds in Hawaii, and they were trying to keep the birds away from this infected pond. You're saying it was an infected pond, and so it was not
in the news.
easy.
It was maybe 5 years ago. something like that.
Okay, about 5 years ago. Okay, I don't remember that, but I'll have to look into it.
I might add something about Hawaii. I work for the sugar industry and research there. And the big island of Hawaii and the Hamakua coast had bubonic plague in the field. And the man in charge was there, and I was there, and they did all this project of getting fleas out of the holes and then checking them. And before I left, they finally found the last bubonic plague in Big Island of Hawaii, in the Hamakua Coast, in 1965. And they checked and checked and checked and checked. But it had been quite a problem before and during World War II among the people who lived and worked in the field.
Yeah, yeah. And so, do you know if it's still eradicated from the Hawaiian Islands?
Yeah, I worked in sugarcane. Basically, because the rodents, 3 different kinds, ate sugarcane. My job was to develop, all my work I did there was for the sugar industry, research baits and poisons that would be suitable there. We probably wound up using warfarin rather than arsenic and gallium sulfate. Yeah, it was, but it was gone. It never showed up after 1965. And they kept that going for 5 years just before I left. And in that 5 years they found nothing. They closed the whole project down. But it was a severe problem in World War II.
Yeah.
I was just wondering how often labs at the CDC actually publish work in normal journals. And if they don't, maybe they have some alternative mechanism where they can spread information that kind of goes through an internal peer review process, but within those own kind of labs. And they get the kind of recognition that you would normally get in a normal journal. If they do stuff like that.
The question was, does the CDC, or Center for Disease Control, publish in the same journals that other scientists do? They certainly do. I'm not sure if it's at the same frequency as other agencies. And if not, are they able to disseminate that information to researchers in a safer, more closed kind of way? I'm not exactly sure. I couldn't tell you the answer to that. I'm not exactly sure. I know there's major spread of information with other research institutions focused on certain diseases. But I'm not sure how much of the information they learn is kept secret. They certainly do a lot of surveillance of infectious agents all over our country and from around the world. And they publish that surveillance data, essentially sequences of influenza virus and sequences of salmonella from an outbreak and things. They just post it online and let researchers see that. Right, you had a question?
Okay. I guess I have a kind of a two-part question. One is, you mentioned that the technology is out there to actually synthesize some of these pathogens, and so I'm kind of wondering, like, if you had to project, how long is it before some guy in his garage would have the tools to actually synthesize this, given the sequence? Mm-hmm. And then secondly, I guess, do you see any risk of bio-research, not specifically on these pathogens, but other bio-research potentially having a sort of spillover effect or unintended consequences on the risk for having this type of pandemic happen?
Okay, first question, can people, if the technology's out there, can the wrong person just build a lab in their garage and make some bioterror agents? The answer is depends on what it is, but for the most part, actually yes. If they have the education and some of the basic